Sendai or Kesenuma, we are in like temporal uh, weather zone. Okay, so let me introduce you about the phytoextraction mechanism of arsenic by Putisvitata. So, okay, so you must know that arsenic has many oxidization state in the environment. It has plus, uh, as an element, it has 0, and it has plus 3 as arsenite, and plus 5 as arsenate, which both arsenite and arsenate are soluble form. But the uh, elemental form and the minus 3 form are not soluble. But uh, prior to be soluble, which is bioavailable for the plant, the Putis vitata accumulate, it the arsenic need to be solubilized from the soil. Then, since this Putis vitata, it uptake arsenate better, much better than arsenite. So it's better to oxidize the arsenite to arsenate. Then it need to be uptake to the Putis vitata. Then the special thing is this fern, it reduce arsenate to arsenite, the arsenic 3. The reason why it reduce arsenate to arsenite, which arsenite is a much more toxic form. So the reason that it reduced to arsenite is still a mystery, but we think that this plant uh, reduce it to a more toxic form and store it in the above ground part might be a strategy of its for its survival to avoid other insects biting. So after reducing arsenate to arsenite, it transported to uh, above ground and accumulate as arsenite. Okay, so uh, if you still remember, I told you that I'm an environmental microbiologist. So I'm also interested in the bacteria. So I focus on the bacteria which interact with Buddhist vitata prior to arsenic phytoextraction. So I focus I focus on these 5 possible plant microbes interactions. The first one is the uh, arsenic oxidization. As I told you, that the Putis vitata likes arsenic 5. So if the bacteria can oxidize arsenite to arsenate, which is much more bioavailable for Putis vitata. And but another thing is actually arsenic 5, which is usually bind with iron, iron, iron become complex and make this complex is insoluble. So another strategy bacteria might have is uh, bacteria. Some bacteria it can secrete hydro4, which is a very strong iron chelator. So with this hydro4, it can uh, withdraw the iron from the arsenate, then make the arsenate available for Putis vitata. That's the second functional we are expected. So the third one is IAA, which is a plant growth promoting hormone. So since you know that arsenic is a very toxic element, so even though it is a hyperaccumulator, the excess of arsenic still become an like environmental stress to it. So if the bacteria can secrete plant growth hormone, so it can help Putis vitata to release from the environmental stress and grows better. So that's the third function we expected. Then we are thinking like uh, this kind of functional bacteria, they may have special interaction with the plant. So we think that that might be the Putis vittata, which uh, extrude some uh, extrudate from the root. So to recruit this kind of uh, microbes, the microbes which can support its phytoexpression. And also, uh, these bacteria might have special chemotaxis, which means to move toward to the plant. So through this kind of plant microbes interaction, that might be enhance this arsenic phytoexpression. So uh, through this hypothesis, we are focused on to find this kind of potential facilitators in phytoremediation, and also to characterize these facilitators and figure out a more efficient method of arsenic phytoextraction. So the approach we used is we tried to isolate uh, this to isolate rhizospheric bacteria from the Putis vittata rhizome, and also we tried to characterize this kind of rhizobacteria. Okay, so let me show you the flow we done. Uh, we tried actually two kinds of arsenic hyperaccumulators. One is Putis vittata, another one is Putis multifida, which is also a hyperaccumulator. So we plant them in arsenic high contaminated soil for a season. Then after that, we take the rhizospheric soil and do isolation. We spread them in the agar plate, and in that agar plate, we put arsenic as a first selection marker. Then after that, we got the colonies with different colonies. So we tried to pick up all the colonies we can with different morphology. So we got a lot of colonies. Then we identified who they are and tried to do further characterization. Then screen the multifunctional rhizobacteria. So from this slide, I will start to show the result. So this is one of uh, the result of the arsenic resistant bacteria we got from Putis multifida. So we got 65 arsenic resistant bacteria from the rhizosphere, and each of them. Then you can see that different number like 8, 4, 8 something. This is the MIC of MIC, the minimal inhibition concentration against the arsenic 3. So we also uh, confirmed the MIC to arsenic 5. So these 65 bacteria, they all resistant to arsenic 3 or arsenic 5 with like diverse concentration though. Okay, so uh, after that, we also try to find the arsenic oxidized gene, which is called AIOA gene. As I told you, the arsenic oxidizing can
Eh,
help the arsenic become
sekelas.
available for Putis
Anjing,
vittata. So we
kelas
choose the
naon.
AIOA gene,
Cesoy,
which is arsenic
tonyok lah.
oxidase gene as another marker. So we use the primer which is specific to this gene and do
Sesuatu kelas
PCR
anjing.
to detect
Kok deken.
all these
Orang lagi kelas anjing
isolates.
malah.
And we found
Malah
that
ini
nine
malah
of them,
bukan
one,
receh
two,
anjing.
three, four, five,
Ditonjokin
six,
pisan
seven,
ku
eight, nine.
dosen teh.
Nine of them contains AIOA-like genes. So we pick up these 9 isolates to further investigation. So
Anjay,
this
online.
pie graph shows the isolate we got from Putis multivita, and we had another get from Putis vitata. So in total, we have uh, 70, 73 isolate from two
Ah,
uh, ferns, and 13 of them
kakak,
contains as
kakang,
arsenic
AA, mau keluar dulu,
oxidizing
mau belajar dulu.
AIOA. So
Hari ini teh ada tugas.
again, these two 13 isolates, we do further characterization. One is the hydrophore secretion. As I told you, that hydrophore can withdraw the ion from the arsenic 5. So we think it is another indicator. So the way we screen the hydrophore secretion is we use two chemicals. One is chrom azure S, and another one is we call HDTMA, hexa decyl trimethyl ammonium. These two chemical combined, they can they will become a complex, and this complex will bind to iron very tightly. The color is blue color. So in the agar medium, we add these chemicals, so the agar become blue. Then we inoculate the isolates like here, and the uh, the hydrophore is a iron, very strong ion chelator, so it can remove the ion from this complex. So become to which make this complex from which make the color from the blue to orange. So from these four, you can see that these four bacteria also all contain hydrophore, and maybe this 20 I think it's 28. No, I think 29. 20 number 29 is the strongest. So this is a qualified test. For the quantified test, we compare with uh, the solution with non-concentration of citrophore. So the third indicator, we confirm the IAA secretion. So IAA is the chemical structure of IAA is like this, and the IAA is made from an amino acid tryptophan. So to screen the IAA secretion, we culture the isolate with tryptophan to let it can uh, produce produce IAA. Then we mix the culture, the suspended culture, with this agent we call Salkovsky reagent, which is uh, H2SO4 with this FeCO3, this kind of solution. So once IAA, which phase to this solution, it will become a very strong iron complex, which this complex shows pink color. And this complex shows the maximum absorbance at uh, 530, 530 nanometers. So after uh, react with this reagent, we measure the solution under the absorbance 530 nanometer and check the secretion of IAA. That is a, quantity qualified test, but to do the quantified test, we also compare with the calibration curve of non-concentration. So through these two method, we combine the arsenic oxidizing ability, the hydrophore secreting ability, the IAA secreting ability as three indicators to screen all the isolate. And we, as a result, you can see here, we got an isolate M318. This isolate contain high hydrophore uh, secreting ability, high arsenic oxidation ability, and also high IAA producing ability. So we we think M318 might be a good candidate for support arsenic phytoextraction. And further, we try to do chemotaxis experiment of M318. Like this is the result. We try to prepare uh, the Butyl vitata root exudate and also uh, several organic acid which usually found in root exudate as a compar comparable materials. And we found that M318 shows very strong chemotaxis toward it moved toward to the root exudates of Butyl vitata. So we confirm that M318 it also contains very strong interact with Butyl vitata. So the M318 it is a Pseudomonas, and after identification we know that it is Pseudomonas mole species. The M318 strain it is a multifunctional rhizospheric bacteria which shows strong arsenic oxidizing ability, siderophore secretion, and IAA secretion ability. And also it shows strong chemotaxis to Butyl vitata. And I will not uh, explain this experiment detail because it is quite complicated, but I will tell you that after the M318 identification, we have done further uh, cultivation experiment in the pot, and we done multi-omics. We use multi-omics approach, which investigate the metagenome, the rhizospheric microbe community, bacterial community in the rhizosphere, and also we check the metabolite in the rhizosphere. And we run a network to show how this bacteria in yellow dot, and also we also check the fungi in purple dot, and the metabolite in pink dot. So we run a network to know this uh, uh, correlation between these three groups and also see their positive and negative correlation. And as a result, we found that one metabolite called putyrosine, it is has been reported as a specific metabolite from Putyrus genus fern. So I think the this putyrosine is secreted by Putys vitata. So the putyrosine has positive correlation with proteobacteria, and the Pseudomonas M318 it belongs to proteobacteria. So we know that rhizospheric microbes and metabolite they correlated strongly, and the putyrosine, which is secreted by Putys vitata, uh, have positive correlated to proteobacteria, and not only M318, but also we have found that the AIOA gene, which is arsenic oxidase gene, as I told you, the AIOA gene was conservative in this group. So we expect the Putys vitata, it secrete putyrosine as a substrate to recruit proteobacteria which contain AIOA to support its arsenic phytoextraction to be more efficient. So we try to utilize this kind of biofunction as an efficient remediation strategy to the real field. Sorry, I I would like to confirm what my my class until 11:10. Okay, I still have time. So um, to a real field. This field is in Miyagi Prefecture. This in this area, and we have two fields. One is in Kesenuma, Kesenuma is this field is close to an abundant mining site, so it contains relatively high arsenic concentration. As you here you can see, the total arsenic content in the K site is 62 milligram per kilogram, and the soluble arsenic is 32 microgram per liter, which the environmental standard is 10 microgram per liter. Okay, and in the field S is located in Sendai City, close to downtown Sendai, but it is a farmland. And the arsenic content is not such high, but the soluble arsenic is slightly over the environmental standard. So uh, we tried this field trial for several years, but we select the planting season from spring to autumn. As I told you, that Putis vittata is a tropical and subtropical plant, but the weather in Miyagi Prefecture, we will have a very cold winter, so that is not possible for Putis vittata. So we plant from spring to autumn. Then during this field trail, we try to do different condition like non-inoculation, which means only Putis vittata planting, and we inoculate the M318, the functional bacteria, to Putis vittata once. We call it PV1. And another condition, we do multiple inoculation. Here we call PV not PV1, PVI. And the extra inoculation is PVII. So during the cultivation, the analysis is we collect the plant every month and we measure the biomass and the arsenic concentration in the biomass. And we also uh, collect the rhizospheric soil to check the total arsenic and the soluble arsenic concentration in the soil. And we also try to investigate the rhizospheric microbes through the AIOA, the arsenic oxidizing gene abundance. And also, we try to know the bacterial composition by 16S rRNA amplicon analysis. So this is the way we try to uh, define the rhizosphere. We try to use nylon membrane bag to define that this is rhizosphere, and the rhizosphere we call bulk. We tried this from 2015.